Hello, welcome to Scholars Hub at Home. I'm Allison Gampel, Associate Director, Alumni Programs at York University. Thank you for joining us today for immunity and COVID-19. First, I want to acknowledge the land York's campuses are built on. As this event is virtual and we are not all gathered in the same space, I recognize that the following land acknowledgement might not be for the territory that you are currently on. If this is the case, please do take the responsibility to acknowledge the traditional territory you're on and the current treaty holders. The website nativeland.ca is a good resource for this. As a member of the York University community, I recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaronto has been caretaken by the Anishinaabeg Nation, the Haudenosaunee Confederacy, and the Huron-Wendat. It is now home to many First Nation, Inuit, and Métis communities. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. Before we introduce our speaker today, I'd like to point you to the numerous stories on our website about alumni just like you. Human trafficking is one of the fastest growing crimes globally, and alumna Michaela Gray Bierman, a friend of mine, is doing everything in her power to support survivors and shed light on this issue. An environmentalist, holistic nutritionist, and yoga teacher, Faculty of Environmental and Urban Change alumna Farah Motani thrives outside of the constraints of a 9-to-5 desk job. Read these two stories and so much more about how alumni all over the world are contributing to positive change. You can visit yorku.ca/alumni and friends for this and so much more. We always like to get to know our audience a bit better with a quick poll before each lecture. So today's question that should appear is, how would you rate your knowledge regarding the topic of today's presentation? A poll should pop up on your screen right now, and I'll give you a moment to respond. Great. Thank you everyone for participating. It's always helpful for us and our speakers to have an idea about who's in our audience and the knowledge that you have about the topic of discussion for today's event. Now, if you need help with the Zoom webinar, do feel free to click on the Q&A button at the bottom of your screen and enter your question. Our team is ready to help you. That same button can be used to submit questions for our guest speaker to answer during the Q&A period following today's presentation. For those of you watching live on Facebook, feel free to submit any questions or comments through the comment section for the video, and the team will send them my way. Please do note that all of your questions and comments are visible to our panelists and staff working behind the scenes, so we ask that you keep your comments relevant and respectful. Today's talk features Jane Heffernan, Professor, Department of Mathematics and Statistics within our Faculty of Science. Professor Jane Heffernan is an inaugural research chair and full professor in the Department of Mathematics and Statistics in our Faculty of Science again. She is co-director of the Canadian Centre for Disease Modelling, leads a national in-host modeling network, and she is scientific advisor to the COVID Immunity Task Force. During COVID-19, Dr. Heffernan has worked closely with Health Canada, the Public Health Agency of Canada, and COVID research networks in France, Australia, the United States, and Canada, including the Field's COVID Task Force. It's an absolute delight and honor to welcome you, Professor Heffernan.
Thank you so much for having me, and I'm very happy to be talking today about some work that we've been doing on immunity and COVID-19. So I'm just going to share my screen.
Wonderful, thank you. I'll see you back after your presentation.
Thank you. Okay, thank you very much for the nice introduction. So today I'm going to talk about immunity and COVID-19. And as was just said, I'm a professor of mathematics and statistics at York University. And so usually what I do is I focus on all my projects on looking at and developing mathematical models to help us quantify and qualify what immunity means, and specifically for today for COVID-19. And so all of my mathematical modeling projects are centered on this key component of immunity. And so what I focus on is this feedback loop that we have between individuals and a population. And so you can think about your immunity at the individual level determines your susceptibility to infection. And then your immunity at the time that you are exposed to a pathogen, if you're going to be infected, that level of immunity also determines how severe your infection will be. Of course, that's also affected by different comorbidities that you might have and your age, and for some pathogens, also your sex and ethnicity can affect your infection outcome. And so what we do is we look at different problems or different questions having to do with immunology and pathogen dynamics in a person. So that means that we work in a field called mathematical immunology. And what we do is we look at the interactions between the pathogen and the immune system, and then we determine how it's being activated. And we looked at the accumulation of antibodies and memory cells after an infection is cleared, or even as it continues if you have a persistent infection. We also do the same thing with the vaccines. So we input a vaccine into our mathematical model, and we look at how the vaccine activates the immune response. And then we look at how that helps generate antibodies and different memory cells in the body that will help stop an individual from being infected, as well as if they're infected, how that immune memory that's gained from the vaccine can lessen the severity of disease, which means that it lessens the pathogen load or the viral load if someone is going to be infected. Okay, so all of those components within the host are important, because when someone is infected, if they have some level of immunity at the time of exposure, then we just said that the level of immunity determines how infectious they can be, because the level of immunity determines what their viral load or their pathogen load will be in the body. And so then that means, because it determines how high the viral load can be, it determines how transmissible someone can be to other people in the population as well. And not only that, we can also use mathematical modeling to look at the mutation risk within a host. And then we can look at the transmissibility of different or new variants of concern that can be developed in hosts and then be transmitted to other individuals. So what we do then is we take this in-host model and we look at how the individuals gain immunity, whether it's from mild, moderate, or severe infection, or from vaccination or different types of vaccines. And then we give an individual some measure of transmissibility at the population level. And then that is also then going to be affected by public health mitigation strategies. And so we look at mask wearing, how that can decrease transmissibility. We look at contact tracing, how that's going to decrease transmissibility because individuals are staying home and so on. And then we look at how all of these different interventions and all of these different levels of immunity and transmissibility are going to affect the persistence of a pathogen or of the infection at the population level. In the in-host model, we also try to quantify the level of immunity that you need in order for you to have, or even your comorbidity status in your age, to determine whether an individual is going to have a mild, moderate, or severe infection, whether they'll show symptoms or not. And then we can track all these individuals at the population level as well, all these individuals who aren't showing symptoms, those individuals who maybe have some mild symptoms, and individuals who have severe symptoms that will need to go to healthcare and maybe to hospital. So all this to say is that immunity is an individual characteristic, but we also have some measure of immunity at the population level, which we also need to study and determine so that we can look at how protected a population is against a pathogen. And that protection can either, if we're talking about, say, importation of a new pathogen, that protection can stop that pathogen from being able to spread when it's first imported. But it also means that if we have a certain distribution of immunity in a population, we can protect the population significantly so that a pathogen that's already in the population can then decrease in prevalence over time and hopefully be eradicated. So all of these things are important with respect to COVID-19. So just a reminder, so what immunity really means is that if someone is exposed to a pathogen, then they can get this infection. And then they can gain some protective immunity. And then that means that if they're exposed to the pathogen again, within a short time frame, they have some protective immunity so that they maybe get an inapparent reinfection. And then much later on, if they are exposed to the pathogen again, then they could get a mild or inapparent reinfection. And what that means is that over time, the pathogen can mutate so that it becomes more fit and so that it has some successful probability of being able to be transmitted between individuals that have some immunity. And so we know that influenza is like that. Influenza has a very high mutation rate. And then so from year to year to year, if you had influenza last year, you might not necessarily have protection from the strains that are going to be circulating this year and so on. And so that means that over time, your effective level of immunity changes because the pathogen is becoming more fit. But at the same time, we also consider something called waning immunity. And so that means that there are some cells in your body, these immune system cells that are generated or after infection or from vaccination or immunization. And these cells and antibodies that are generated from that can also decay over time. And we know that this happens, for example, for pertussis. So that's a reason why, and that pertussis is also called whooping cough. So we know that immunity gained from infection from pertussis and vaccination against pertussis, that immunity can wane over time. And then what we do is we look at what this waning rate looks like over time, and we use that information to determine when new booster doses of the vaccine are needed. And so if you look at your booster dose schedule for pertussis, you'll see that there's a certain number of doses that are needed when you're between 0 and 5, and there's another dose a little bit later and another dose a little bit later, and so on. We do know that for common cold coronaviruses, the common cold, which is also a coronavirus, that immunity wanes over time. And there's some indication that moving from a full level of immunity to full susceptibility can happen between 3 to 9 years for common cold. And so while we don't know what the waning rate is going to be for COVID-19 or SARS-CoV-2, we can use this information from the common cold coronaviruses and some studies that are coming out on looking at some waning in different population cohorts to feed into our mathematical models so that we can use that information to project what COVID-19 will look like in the future. So we can fit mathematical models to the past using all this information, and then we project what they're going to look like in the future so that we can get an idea of what these distributions of immunity really look like at the population level so that we can quantify what protection will look like. Okay. So this is a basic mathematical model at which all mathematical models for COVID-19 are based on. And all it does is it takes the entire population and it splits the population up into people who are susceptible, people who are infected, and people who are removed. So either recovered from infection or who have died from infection. And So this is a very simple model. The COVID-19 models are much more complex, but you can see that what happens is the susceptible population can decay over time, the infected population increases, and the recovered or removed population thus increases. And you hope that that's mostly people who are recovered versus people who've succumbed to the disease. When you incorporate public health mitigation strategies into the model, you won't just have one peak like this. You'll have a lower peak, and then it will decrease. And as people relax their behavior, or as public health mitigation strategies are relaxed, then you can get a rebound. And then later on, and then there could be more public health mitigation strategies being implemented, and then that will go back down, and then you'll have some relaxation, and you can have another one. And so you can get, these periodic peaks of like these increases and decays, and these are related to public health mitigation strategies and our individual behavior as well. So mathematical models also need to incorporate some decision-making capacity in all of our individuals. And what this really means is that essentially, when you're looking at a specific pathogen, the pathogen is going to try to infect a certain number of people in the population. And if we implement public health mitigation strategies, and then we have relaxation of behavior, essentially we'll get these rebounds of the pathogen for a long time until there's a sufficient level of immunity in the population such that the pathogen will die out. And that usually is about at the same level of infection as or these removed individuals here, except that it will happen over a much longer time period. The reason why we do that is so that we can bring in a vaccine so that this level here is actually decreased, so that we have some level of immunity earlier on that will bring this level lower. Okay. So when we talk about eradicating a pathogen or getting rid of a pathogen in a population, this is all related to this key concept in epidemiology called the basic reproduction number, or the reproduction number. And you've probably heard about that in the media. What's the current reproduction number? You can go on different websites or different dashboards for COVID-19, and you can look at what the RT is, or the current R number. And what the R number is the number of secondary infections produced by an initial infective in a totally susceptible population. So this is the basic reproduction number, which is at the very beginning of the epidemic. And then we have these RTs that currently go through over time. They change over time depending on the mitigation strategies and the level of immunity that's being gained in the population. And what we want is for that reproduction number to be less than 1. So that, which means that an individual who's infected on average infects less than one other person in the population. So if I'm infected, this is me, and I infect some other disease modelers in the mathematics and statistics department here. Here I've infected 2 individuals, though I had contact with all of these individuals, say, during my infectious period. So my reproduction number is 2. But if you have lots of other individuals in the population and they didn't infect anybody when they were infected, then on average, that number is going to be less than 2 or less than 1. And that's what we want. We want that number to be less than 1 so that the pathogen will decay in the population. Okay. So that reproduction number is actually really important, that initial one, and it helps us determine the fraction of the population that we need to have protection against the pathogen in order for the pathogen to be eradicated. And so that's what this is. This is the fraction of the population that needs to be protected, and it's given by 1 minus 1 over that initial reproduction number. And so if that initial reproduction number is 2, then that means that 50% of the people in the population need to have immunity against infection in order for the entire population to be protected. If that reproduction number is 12 to 18, then 91 to 95% of the population needs to have immunity against infection in order for the entire population to be protected. And you can see here, so I'm listing a lot of these reproduction numbers here for all these different pathogens. And this is SARS from 2003, and it has a similar reproduction number to the current SARS-CoV-2 or COVID-19 between 2 and 5. And so if we look at the historical strain for COVID-19, it has a reproduction number of a little bit between 2 and 3. And so we need 50 to 66% of the population to have immunity against infection in order for the entire population to be protected. However, we know that as the new variants of concern have been coming into the population and increasing in transmission, these pathogens have higher fitness than the historical strain that we first started COVID-19 infection out with. And the current Delta variant is 2 times the fitness of the historical strain. And so what we're looking at is a reproduction number between 4 and 10 instead. And so we might want to look at a reproduction number of about 5 in order for us to look at what that protection needs to be in the population. But I'm going to say here that these are requirements of protection against infection, but we know that immunity can wane over time. We also know that the immunity that we gain from infection and from vaccination doesn't necessarily give us perfect protection against infection. It might give us protection if we're infected. It also gives us protection against getting severe infection. And so then what we need to do is we need to increase these numbers even higher. And I'll give you an example on the next slide. Okay, so suppose that our initial reproduction number is 5, and so that would be for the Delta variant. If a vaccine is 80% effective in generating neutralizing immunity, that means it's 80% effective in stopping you from being infected if you're vaccinated, then we need 100% of the population to get a vaccine. Okay, or 100% of the population to be vaccinated plus have gained some immunity from infection, given that our vaccination program started about 10 months after the historical strain started to be circulating in the population. If a vaccine is 95% effective in generating neutralizing immunity, protecting you from infection, we need 85% of the population to have some immunity neutralizing or to go get the vaccine, or 85% of the population to be vaccinated plus have some people have been infected. So we're gaining immunity from infection and from vaccination. But what we know about the current vaccines is that while they can be protective, they can protect individuals against infection, you can still get infected. There's a small probability of infection, even though you're vaccinated. And then you have a protection against severity of disease. So these numbers are actually going to increase such that when you have a reproduction number of 5 for the Delta variant, more than 100% of the population needs immunity in order for us to get rid of the pathogen. And that's why, and that's not including, so that's just looking at immunity. And so then that means that in order for us to get rid of infection, we can't vaccinate more than 100, we can't give immunity to more than 100% of the population. And so what we need to do then is we need to maximize our level of immunity in the population. And we need to also add in public health mitigation strategies like social distancing and mask wearing and so on, so that we can get that eradication to happen, because we want that reproduction number to be less than 1. And the reproduction number is affected by your immune system, and it's affected by the transmissibility between individuals. And that's why mask wearing and social distancing, working from home, those things are all going to help get that reproduction number less than 1 so that we can hopefully get rid of the pathogen. And then that means that we need to also consider things like vaccine hesitancy. So we know in Canada that vaccine hesitancy in different age groups ranges between 10 to 25%, and adults make up roughly 80% of the population. So we need all adults to be vaccinated in the population, or all individuals who are 12 plus to be vaccinated. And then plus we need public health mitigation strategies for us to get that reproduction number to be less than 1. Okay. So that simple threshold model of 1 minus 1 over R0, it doesn't incorporate age structure. It doesn't incorporate different contacts that individuals at different ages have at school, home, work, or other. It also doesn't consider, because it doesn't consider the age structure, it doesn't really consider the age distribution of the vaccine over time, and it also doesn't incorporate the slow start and then large increase in getting the first dose in Canada, and then the greater focus of getting the second dose over the summer. It also, that simple model doesn't incorporate differences that I talked about before, like looking at mild, moderate, or severe infection, how immunity is gained from those. And it doesn't differentiate between the type of immunity that you get from vaccination versus infection, or different types of vaccines, like the mRNA vaccines or the adenovirus vaccines or protein subunit, different types of vaccines that are currently in clinical trial versus vaccines that are already being used in the Canadian population. And that model also doesn't consider waning immunity. So what we've done is we've used mathematical models to look at how immunity is generated in an individual. So we track different cells and pathogen in the body, and we look at the interactions and how it activates the immune response. And then we do that for the vaccine too. And so what we do is we look at the virus life cycle. So here's a SARS-CoV-2. We have the virus attaches to the cell, gets input into the cell, and a bunch of things happen within the cell. And then the cell becomes like a factory for making new virus particles. And so we have one that goes in, and then we have virus particles that get released. When all of this is happening, the immune response, so it's different types of different components of the immune response start to be secreted by these infected cells, which then activate different other components of the immune response, like T cells. And then the T cells help activate other components of the immune response, so that you can get this big immune response that incorporates B cells and antibodies and CD4 T cells and CD8 T cells, so that the B cells and antibodies can get rid of the virus, and the CD8 T cells can kill the infected cells. And you need all of that to happen in order for an individual to recover from infection. And then when the individual has recovered from infection, we get this accumulation of B memory cells and T memory cells, which then can be activated at a much faster rate when an individual has been re-exposed to the pathogen. And the vaccine works in a very similar way. It doesn't infect cells necessarily, but it does help induce secretions of different components like cytokines and for the immune response, which then activate and excite the cells in the immune response, which then make these B memory and T memory cells. So here is a model that we've actually developed for an adenovirus vaccine. We're looking at 1 dose versus 2 doses, and this is data from an AstraZeneca clinical trial, where you have 1 dose here and you look at 2 doses. And they've been, so this is looking at an IgG response that's being measured. And this is looking at interferon gamma. So these are 2 components that they'll measure in the clinical trial. And then we develop a mathematical model that incorporates interferon gamma and IgG into the body. And then we fit our mathematical model, our equations, to this data. And then what we can do is we can look to see what our antibody level is going to look like at the end of infection, and what our T helper cell population is going to look like, so that we can get an idea of if someone's exposed again to the pathogen, or exposed to the pathogen after vaccination, what their immune memory will look like. and how that changes depending on the number of days between doses of the vaccine. And so here's an example of what we get from our mathematical model. So we've given 1 dose of the vaccine at day 0, and then we give the second dose either 28 days later, or 40 some days later, or so on to 70 some days later. And then we can see what this looks like if we look at this at the T helper cells. So after 1 dose, we have, and this is in arbitrary units, we have some level of T helper cells, and you can get some decay. And then there's the second dose of the vaccine, and then you get this boost in T helper cells, and then you can get some decay over time. We do the same thing for plasma B cells. So after the first dose, and these plasma B cells are related to antibodies. And so we get this increase in plasma B cells, they help make antibodies. And then in the second dose, if it's 28 days later, you can get this jump in plasma B cells. And if it's 70 some days later, you get this jump in plasma B cells. And so these are the activated cells, the T helper cells and the plasma B cells. And then what happens is these decay over time. These cells, some fraction of these cells become memory cells. And then the memory cell population increases in the body. And those memory cells can live for a very, very, very long time. And your body also practices something called homeostasis. It wants to keep your T cell and your B cell populations up at a certain level. And so those memory cells can also divide over time as well to make sure that your T cells and your B cells are maximized in your blood. Okay, so what we do is we look at mathematical models of this in-host modeling to also look at mild, moderate, and severe infection and these vaccine outcomes. So then what we do is we put this into a population level model where now we're tracking different levels of susceptibility. So these are people who are 100% susceptible to COVID-19. These people have some immunity against being infected, but they also have some immunity against severe disease. And these have a higher level of immunity against infection and a higher level of protection against severe disease. And then we have individuals here that have full protection from infection. And so we allow people to get mild infection and they get some level of immunity. If they have a moderate infection, they have some level of immunity. And if they have a severe infection where they've gone to the doctors and maybe to the hospital, they get a higher level of immunity and full protection. And the probability of having mild, moderate, or severe infection depends on the comorbidity status in the different age groups. So we model 5-year age groups in the population. We use the Canadian population distribution, and we also look at different provinces and territories as well. And we use that information to look at mild, moderate, and severe infections so that we can get an idea of what healthcare demand will look like for ICU and ward beds, as well as having an idea of what these asymptomatic infections, or these individuals that don't show symptoms, how many of them are in the population, given a certain, if we have a reported case, how many other infections are there that aren't reported? Okay, and then that depends with contact tracing as well. So we have this distribution of immunity that we get in these individuals who are in these S classes. And then we also give individuals the vaccine. They come from these S classes and they can have 1 dose of vaccine or 2 doses of vaccine and so on. And we give these vaccinated classes also some protection against infection and some protection against severe disease if that individual does get infected. So using this mathematical model, we fit this model, we inform the model from all the in-host modeling that we've done for mild, moderate, severe disease and all the different vaccines that we've looked at. And then we've informed the model, given it this structure. And now we fit this model to the past COVID-19 infection history in the population. And given all of the different public health mitigation strategies and lockdowns and school closures and things like that. And we then project forward to see what the fall will look like. And so here is an example of something that we see. So we're looking at different age groups here over different levels of immunity. So these are So see, we have dark purple, a lighter purple, a lighter purple, and a very light shade of purple. And so these are individuals in different age classes that are in that dark purple. And these individuals, we say, have full protection from infection. And so you can see that there's a very small level of these individuals. And then the vaccination campaign starts to be ramped up. We get this big increase. And now we have lots of people with 2 doses of vaccine. And some of these people were infected and recovered from COVID-19 as well. These are individuals that have 1 dose of vaccine, and some of these individuals are recovered from infection where their immunity has waned. These are individuals that might have 1 dose of vaccine, but coming from lower levels of immunity, and they have a certain level of susceptibility. And then these individuals in the red class here, they're individuals that are totally susceptible to COVID-19. You can see how that's decreasing over time as the immunity is gained from infection and from vaccination in the population. And so if we look at the top of the blue curve here, so this gives us an idea of what the distribution of immunity might look like. I don't know for sure, but this is some picture of what the distribution of immunity might look like in the Canadian population. And we are about here in the current timeline. So we fit the data up until this dashed dotted line. And now we're here, and we have individuals that have a high level of immunity, a lower level, and a lower level. And then these individuals have full susceptibility to COVID-19 in these different age groups. And so what you can see is that our population doesn't sum to 1. And that's because the individuals that we don't have are individuals who are currently in those infected classes, who are infected and not transmissible, or who are infected and transmissible and maybe in the hospital and so on. So by the end of the summer, before the beginning of September, we find in a lot of different scenarios that we're looking at with different rates of waning immunity and susceptibility and different vaccine combinations that we look at, between 60 to 80% of the population will have some level of protection from infection and severe disease. So the example that I'm showing you here, by the beginning of September, this example has about 70% of the population has some immunity against infection and severe disease in the population. In some of the scenarios we look at, this is as low as 60%. And in other scenarios, we see that this is as high as 80%, but we don't see any higher. And that makes sense given the levels of vaccine hesitancy in the Canadian population. But what that also means is that we will see a resurgence in the fall. And this resurgence, depending on the relaxation behavior of individuals at the beginning of September, what that looks like can be pretty big and can be bigger than what's been previously experienced in the population. But if we're actually in the next 2 weeks able to really do well in making sure that those case numbers are really low, then we can also see that this fall wave could be almost non-existent. It could be really, really small. So it really does depend on what individuals do in the next 2 weeks, how big this wave in the fall will be. And I can't say for other modelers, but in my models, I've seen that school opening in September doesn't really have much effect on what this fall wave will look like. So it really just depends on what we do in the next couple of weeks in the summer, and that's going to determine what the fall wave will look like. School opening doesn't have a big effect on the magnitude of that fall wave. So given our now knowledge with respect to immunity, we know that from infection and from vaccination, the Canadian population has some immunity between 60 to 80%. But that's not full protection from infection. That's protection against infection and severe disease if infected. And we know that that level of immunity then determines what the fall wave will look like. But additionally, that also means that we need to consider public health mitigation strategies. And so The severity of the fall wave is also going to depend on our behavior for the next couple of weeks, as well as in September, what our social distancing behavior looks like. Hopefully it's not relaxed too much, and that we keep on wearing those masks, as we do know that they're effective in staving off infection. So I look at lots of different things in mathematical immunology and in epidemiology that have to do with immunity. And this work is in partnership with lots of collaborators that I've listed here, and it's funded by lots of different funding agencies and collaboration networks as well that I've listed down here. Thank you so much, and I'm happy to take some questions.
Dr. Heffernan, thank you so much for being here and sharing that wealth of information that I'm just, trying to process and figure out. I'm guessing in the audience here, we're all trying to figure out how to process it and how it applies then to our own lives and situations. So thank you so much for sharing it. So for our audience, if you have questions that you'd like to ask, please do feel free to type them into the Q&A function, and I will ask them on your behalf. And we have some questions that have already come in, so I'll start with those. The first question is one that's actually quite specific, and it goes back to a slide that you showed earlier, where the question was, there was a list of showing the various illnesses, specifically measles, SARS, and under the section of transmission, there were some that were called airborne versus airborne droplet. Is there a difference between those two, and could you explain that?
Yes, so they're different. When you cough or when you sneeze, you expel droplets and aerosolized particles. And when we look at the transmissibility of an individual, we look at something called an infectious quanta. And the quanta might need to be big. It might need to be a big clump of virus that's in a droplet, or we might not need a lot of different virus particles, which could be in an aerosolized, very small, fine particle. And so when we look at these different droplet and particle components, people in fluid dynamics, what they do is they study these cough and sneeze plumes between individuals that don't wear masks and people that do. And then the droplets, they can only travel a certain distance and then they fall down to the ground. The aerosolized particles can float into the air and span a much greater distance. And so that's why we have these social distancing measures of 2 meters or 1 meter, depending on where you are in the world. But that has something to do with whether something is transmitted by droplet or if it can be aerosolized.
And so for the COVID that we're facing and the Delta variant, do we have a sense of which it is, droplet versus airborne?
It's a combination. So
okay.
In the beginning, people were focusing more on droplet, and things have shifted to be looking at aerosolized transmission.
Okay, thank you so much for that. Okay, so let's talk about kids. I know I myself have kids that are on their way back to school, and you mentioned that the kids going back to school actually won't have all that much impact on the fourth wave, yet the kids, and I think in Ontario, were learning from home, if you call it learning, for a very long time. So can you just tell us more about this whole idea of kids and how kids can be in community and perhaps unvaccinated, more susceptible, but yet that might not impact a fourth wave?
Yeah, so something, remember I look at those mild, moderate, and severe infections, and the probability of having a mild, moderate, or severe infection depends on the comorbidity distribution in an age group. And the comorbidities, accumulate as you get older. So they're much, when you look at comorbidities that are going to increase your probability of severity of infection for COVID-19, those are much higher in the elderly than they are in children. And so there will be infections in children that are mild or inapparent, that are asymptomatic. But we also know, remember I was talking about viral load. So when you have a mild infection, it means that you don't have as high a viral load as you do if you have a severe infection. And so you're less transmissible. And so while children can be infected with these mild or inapparent infections, like So one of my sons had COVID this past year, and you wouldn't know, and the rest of the family, none of us got it, and it was in part because his infection was so asymptomatic that he wasn't expelling much virus, and so, or maybe even any virus really, and so there was no transmission in our household, and there was no transmission amongst his friends as well. So he got it from another connection outside of school. And so that's really what it is that while there's a lot of there could be mild infections in schools, there's not a lot of transmission that's happening between children, especially if they're wearing masks and they're practicing social distancing in the classrooms. And if they're outside, they're running around and in an environment, there's a lot less transmission probability happening while they're outside at recess as well.
Thank you so much. That's incredibly helpful. Okay, I'm going to ask another question that's coming in from Paula. Paula says, and I'll read it, Dr. Heffernan, in 2019, there were numerous measles outbreaks in Canada. Were you involved in any of the modeling related to these outbreaks? Even if you weren't, how can mathematical modeling be of use when vaccine coverage is waning in a population due to lapses or lower vaccine uptake?
Yeah. So measles is one of the viruses that I focus a lot of my work on. And a reason why I do that is because there is a lot of belief in the research communities and in the population in general of how immunity is gained from infection versus vaccination. And there's some it's been believed for a long, long time that once you have immunity against measles, you have it for the rest of your life. But there's been some indication in some studies that there could be a small decay over time in that immunity, and a small level of waning immunity, given a reproduction number of 12 or 18 for measles, which is very high, can mean that we can have some asymptomatic infections happening in a community if measles is imported. And so then that means that if you see a couple of measles cases, there could be some asymptomatic cases that are also happening. But because measles is a reportable disease, there aren't a lot of, I don't expect that there's wide circulation of measles in a population. But I do, yes, I look at the outbreaks that have happened in Canada and the United States over the past few years to see if I can get a better idea of how to quantify the effects of waning immunity against measles in a population if immunity wanes. There's still a lot of controversy around that.
Awesome, thank you for that. When we speak about the variants of concern, and how do the variants, and I guess particularly now the Delta variant, and do we think other variants are going to emerge that are going to be of concern? How do those affect immunity and herd immunity?
So the mutation rate for COVID-19 or SARS-CoV-2 is very low. It's much lower compared to, like, say, influenza. But the issue that we have is that this is infecting the entire world. So you have a lot of people who are being infected, and because you have lots of people who are infected, you have a higher probability of generating these variants of concern. And some of these variants of concern are actually generated in severe infections where they've tried, different treatments, which have kind of selected for the fitness or different mutations in the receptor binding domain or in the spike protein in general. And so it's totally possible that new variants of concern can emerge into the future because the entire world is being infected, and one of these new variants could emerge in Canada. It just really depends, and it's all based on probabilities. And just because the entire world is being infected, there's a higher probability. There's a small probability of generating a mutant, but when you have all these people being infected, it's very certain that new mutants can be generated.
Right. So then with regards to these booster vaccines that I believe some countries have actually started implementing booster vaccines, what can we expect, or what should we be doing here in Canada?
So some of the studies that have looked at immunity in different individuals at different ages from the vaccines, it's been shown that there's a decay in immunity, a much stronger decay in immunity in the elderly. Someone who's older could have 2 doses of vaccine, but there's a lower level of effectiveness of the vaccines in the elderly, as well as the immunity can decay faster in people who are older. And so we do see a significant level of decay in frail elderly individuals. And so the idea in discussions with different levels of government and all the different researchers and modelers and public health officials, that maybe there will be a focus on having a booster campaign for at least individuals who are older in the Canadian population.
Thank you. And do you think coming soon, I've got an 11-year-old. Do you think coming soon, children under the age of 12 will be receiving the vaccine?
Yeah, I think that there is a vaccine that's in clinical trial in younger children, and I'm waiting too, because one of my sons is younger than 12, and one of my sons is older than 12. So one could be vaccinated and one can't, and I'm just waiting for that vaccine to come out too, so that he can be vaccinated. So even though, like I said, that children are less susceptible or less transmissible, just at least having that extra protection in the population can also help protect, the more vulnerable children as well, and it will just, give some more sense of security for everyone to have all of their children vaccinated.
Absolutely. I know I'm counting on mine, her 12th birthday. I've also got those 2 scenarios. So you have done such a wonderful job of sharing so much information and so much complexity in a way that's been so easy to digest and understand. So thank you so much for doing that. We don't have further questions from, actually, I do have one question that just came in. I'm going to take this as our last question, and then I'm going to ask if you have any info about your department, math and stats, and what other cool things you work on. I mean, now these days it's super exciting. But I am going to read this question that's come in from the audience. The question is, maybe my question is out of topic, but I usually wonder why large populations in Africa, especially countries in Eastern, Central, and Western Africa, did not have lower infection, hospitalizations, or do not, I can't read it, do not have severe infection, hospitalization, or death. Can it be related with some pre-existing immunity or the way that people live? And this person goes on to say, I know that countries in Africa are not having hard lockdowns or mandates to wear masks, and many people are living normal life, and there doesn't seem to be severe infection or death. So if you could speak to that, that would be fantastic.
Sure. There are lots of different factors that can contribute to that. One is different lifestyle. If you look at the mobility patterns of some individuals in areas of Africa versus in North America, there could be less distance that individuals travel away from their home during the day. But then you also have workers who are going to travel very far distances for work. So there are some things that researchers are looking at to look at these differences in mobility. Another is if you look at the distribution of age in a population. So I've focused a bit on my research on looking at these comorbidity status and how that affects the severity of infection. And so in populations where the age distribution is skewed to younger age groups, or if the top age is lower compared to other age distributions, you would expect to see less severe cases in a population on average, just because of the age distribution and these comorbidities. Of course, we also need to take into consideration the healthcare capacity, though, of all the different countries, and the ability for the healthcare system to be able to contact, trace, test, and report and hospitalize. And so there could be in some of these other countries some issues with respect to how the data is acquired, how the data is reported. So there are lots of different factors, but I think that those are all important factors to consider when with that question.
Fantastic, thank you. I'm going to ask a final, I said that was a final question, but I have one more final question that came in from Merdad, who thanks you like I do for a great presentation. They're wondering if COVID became seasonal like the flu, what mitigation strategies would you recommend?
So we know that COVID-19 can give some severe infections to some individuals, and we know like flu, influenza can do that too. They're not the same, they're different, but you can have similar public health mitigation components or strategies for that. And so depending on, the evolution or the mutations of the virus and the waning rate of immunity, you might think that if COVID becomes a persistent infection, kind of like a common cold in the population, but a severe common cold, you might see some vaccination campaigns, maybe yearly, for some different cohorts, or because the mutation rate is pretty low for COVID compared to flu, and if we're able to keep our immunity up, because there's maybe some relationship between this coronavirus and the common colds, that maybe this will just become another common cold, and we don't need to worry about some future public health mitigation or vaccination campaigns.
Fantastic. Well, that will conclude today's conversation. So, Dr. Heffernan, thank you very much for the work that you are doing and continue to do. Did you want to tell us a little bit about any other cool things happening out of your department?
Sure. So I'll say a couple of things. So one is everything that I talked about today is mathematical modeling. It's not exact science, and it's not exactly biology, and it's not always observed. And so the modeling results that I showed you, while they can give you an idea of what is actually happening, they're not the truth. However, I also wanted to say that we have a degree program in our department, Math and Stats, called Mathematical Biology. It's the only one in Canada. And if individuals are interested in becoming mathematical biologists like me, where maybe they would focus on answering questions to do with animals or plants or disease or other things in biology, looking at patterns or even how a zebra gets its stripes and how a leopard gets its spots or things like that, then please reach out. I'm the director for the mathematical biology program here at York. And if you have individuals in your family or friends that are interested in all of this disease modeling that's been happening over COVID and want to learn more, please check out our program, and you can feel free to email me any questions.
Thank you so much. That sounds like a really cool thing to study and so important. So thank you for being here. Thanks again. And I have some concluding remarks for our audience. So I'll let you turn off your camera and wish you and your family good health and be well.
Thank you very much, and have a great day, everybody.
Thanks. Okay, so before we say goodbye to our audience, we really want to thank you for taking the time to be here, and thank you for your really thoughtful questions. I do apologize that earlier in the conversation, our poll had some technical difficulties, but we would still like to know if you could answer a poll that will pop up on your screen, and it should pop up this time. So how would you rate, there it's there now, how would you rate your knowledge of the topic of today's presentation following this insightful discussion? So please take a moment to respond. Perfect, thank you all so much. So join us in September for more Scholars Hub at Home. It'll take place every Wednesday at noon. On September 15th, we bring to you Waking Up the Medicine, Instilling Anishinaabe Knowledge into History. And then on September 22nd, join us for Privacy in Virtual Classrooms and the Key to Equity and Inclusion in Online Classrooms, Lessons from the COVID-19 Pandemic on September 29th. For information about these and any of our other events, please visit our website anytime at yorku.ca/alumni and friends. Thank you all for being here, and please be well.